I am very excited to introduce Teresa Davoli, who is an associate professor in biochemistry and molecular pharmacology at NYU. She has done a lot of groundbreaking research in aneuploidy, including a very important finding in 2013, where she and her group found that the patterns of copy number losses and gains are strongly associated with the number of tumor suppressor genes and oncogenes in cancer. And as an aside, we use this result in my lab all the time, and we always refer to it as the Davoli score. She has won a number of awards for young investigators, including from the Melanoma Research Alliance, Breast Cancer Alliance, and Pershing Square. So please join me in welcoming her.
Okay, thank you. Right, thank you for coming to the room. Thank you all for people on Zoom for being here. Is the audio good? Audio, video, everything is good. Okay, great. And thank you for the kind introduction as well. Okay, so today we'll talk about aneuploidy and chromosomal instability. I will tell you a few stories from my lab. And actually, my talk was quickly revised right before this seminar, based on conversation that I had today, this morning, with students and postdocs and PIs. So students, don't take that as an advice. Never change your talk last minute, but really that inspired me on sort of showing and changes likely in order to be even closer to what you guys do. So these are my disclosures. And yes, so the concept of aneuploidy stems from the fact that there are billions, quadrillions of cell divisions in our body. And so in some cases, in a few cases, the chromosome segregation in the two daughter cells fails, leading to chromosome gains or loss after the fault in mitosis. And this can lead to different type of outcome, including chromosome gains, chromosome loss, presence of micronuclei, lagging chromosomes, additional focal aneuploidies, also DNA damage, DNA breaks, and several other consequences. But today, the focus is really aneuploidy, which is already defined, is the presence of gain or loss of chromosome. And as you know, at the organismal level, it's very rare. It's the first cause of abortion. And so with a few exceptions, is incompatible with development. However, in cancer, as you know, the aneuploid state is extremely frequent. We are talking about more than 90% of solid tumors that are aneuploid, as well as a good amount of leukemias, slightly less, but on the order of 50–60%. So aneuploidy is not only very frequent as a state, as a karyotypic state in the tumors, but it also carries with it the fact that specific chromosomal gains and losses are particularly frequent across tumors. And so this is a figure from a research paper, actually from Alison, who's here, and Rabin Dana-Farber, where I think they beautifully plotted the frequency of events, of somatic events in tumors. And as you can see, the top, I believe 24 of the top 25 most frequent events in tumors are aneuploid. And so 20q gain, 7p gain, 8q gain, 17p loss, and several others. So really, with the exception of p53 mutations, which is the king here, the most frequent somatic events, the next most frequent alterations are really copy number events. And so several labs, including the lab of Alison Taylor and others, have studied this phenomenon for years. And what we know now is that aneuploidy, while being detrimental for, as I said, for embryonic development and also in general for tissue homeostasis, in certain contexts, the presence of aneuploidy and chromosomal instability has been shown in mouse models and other settings to promote tumor formation. In humans, perhaps the best case, the best example of aneuploidy as a tumor-promoting event is represented by a very rare syndrome that is called MVA, mosaic variegated aneuploidy. There is actually a center here at Columbia. It's a very rare syndrome, but here at Columbia, there is a center for this disease. So people with this disease inherit in their germline mutations in genes that code for proteins important for mitotic segregation. And so they have a certain level of random chromosomes missegregation in their body that leads to the mosaic accumulation of aneuploid cells. And this condition is very, very highly associated with increased risk of solid tumors, especially. So this is in a way the genetic close genetically between chromosomal instability and tumorigenesis. And so, of course, key questions in the field are to understand whether and how aneuploidy drive cancer, and of course, ultimately use aneuploidy as a biomarker for risk and for therapeutic response, as well as potentially a therapeutic targets. As it was already previously alluded to, cancer aneuploidy is highly chromosome specific, so not all chromosomes are gained and lost at the same rate, but there are very specific patterns that are observed across tumors. So, for example, chromosome 9 is frequently lost. So this is a pan-cancer overview of solid tumor. Chromosome 9, especially in NP, is frequently lost. Chromosome 10 is also frequently lost. Chromosome 7 is frequently gained, as well as 8q, chromosome 20, and others. In addition to, and so this chromosome specificity is a very important feature, and we and others contributed to try to understand what are the features, what are the characteristics of these chromosomes that make them so frequent across solid tumors. Another important feature of aneuploidies is that certain aneuploidies happen early in tumorigenesis and also persist. So we can see this phenomenon by looking at single-cell DNA-seq data. These are data from Nick Nevins and others, where you can see that in these breast cancer patients, where each row is a cell, and here are the chromosomes, you can see that even in the most unstable and very heterogeneous from the genomic point of view tumor, you still find events, for example, in this case, a peak on chromosome 8, on chromosome 5, on chromosome 1, that are present and persist across the cells of the tumors. So this would suggest a strong selective pressure to keep the chromosome or to reacquire the chromosome. Similarly, in other studies, it's been shown that at a distance of several years or decades from a primary tumor to a metastatic tumor, in this case a breast cancer, same aneuploidies were persisting in the tumor between the primary and the metastatic setting. So ultimately, one additional reason why we care about aneuploidy is the fact that often it's been shown by our group and others that it can be a strong predictor of progression, so of cancer risk in the case of pre-malignancies, or also of survival as well as therapy response. So these are just some examples where 9p loss, for example, predicts a progression from oral pre-cancer to cancer. 18q loss predicts poor survival in colorectal cancer, and 1q gain predicts poor survival in endometrial cancer. And so overall, my lab and others are really trying to develop computational experimental tools to understand how the genotypes of the aneuploid cells determines the cell states and ultimately the cancer hallmarks that we all know. And so these are the questions that I already mentioned. So what are the biomarkers? How can we use aneuploidy as a biomarkers? And can we ultimately target aneuploidy for cancer therapies? And so today I will give you two examples of stories of approaches that from my lab. One is about it starts from a 90 stories, so it starts from 90 laws as a biomarker and a predictor of therapy response and immune evasion in certain tumors, and then it's also expand to other aneuploidy patterns in the same context of tumor microenvironment. And the second part, I will talk about how our CarioCreate method and how we use it to model one specific type of aneuploidy. So to start for the first part, I don't have to tell you that the interaction between cancer cells and the immune microenvironment is extremely important at all stages of tumorigenesis, from the pre-cancer to cancer evolution to the metastatic stage. So tumor cells have to continuously to resist the recognition and killing by the immune cells, especially by the CD8 T cells, which are the masters of anti-tumor immune response. And so these are just some mechanisms, some common mechanisms of resistance by which tumor cells become resistant to the immune system, including decrease the response to interferon gamma, decrease MHC-1 presentation, and others. And so this study, in a way, this project, in a way, started from an observation that we made years ago from our lab and others, where we saw that the overall level of aneuploidy in tumors was associated with an immune cold tumor microenvironment. How did we measure the microenvironment of the tumors? We used an RNA-seq-based method to estimate the level of cytotoxic markers in the tumor microenvironment. For example, genes like here, granzyme perforin and others, mostly associated with cytotoxic CD8 or NK cells. So calculating an overall cytotoxic immune score for each patient, and then correlating that with different genomic features. Finding that the overall level of copy number, or SCNA, was associated with a decreased level of this cytotoxic immune score, and so to an immune cold microenvironment. And so the one of the tumor types where this was particularly strong was head and neck cancer. And so we were lucky to start a collaboration, actually started during the pandemic. So this collaboration went on for years without having even met in person. My collaborator from the West Coast, that was an interesting experience. But anyway, so this project was focused on head and neck cancer, which is an important, it's a quite prevalent type of tumor. It comes in different forms, including HPV positive and HPV negative. In this case, we're only focusing on HPV negative patients, so not associated with this virus. In terms of immunotherapy response, about 15% of patients respond to pembrolizumab or other immune checkpoint inhibitors. And there is really not a good biomarker for response, in the sense that the level of PD-L1 or the tumor mutation burden are not good predictors of response. And so we put together different data set and different type of analysis, including oral precancer samples, as well as oral cancer tumors and cancer cell lines, and analyze and stratify this patient in different ways based on the copy number, and also based on an assessment of the tumor microenvironment, in some ways based on an immune score based on RNA-seq, and in others also based on IUC. And ultimately, what we observed in a nutshell in one slide is the following, is the fact that, so here I'm plotting the frequency of events, so the frequency of gains and losses in oral cancer patients. So as you can see, there are certain events that are more frequent than others. For example, 3P loss and 3Q gains are very frequent, 8Q gain as well. And now here at the bottom, I'm showing not the frequency anymore, but the association with the cytotoxic immune score. And so a high value on the y-axis would indicate an association with a high immune score, so immune hot tumors, and a low value would indicate an association with immune cold. And so in this landscape, it stands out 9P, where 9P loss is associated with immune cold, and 9P gain is associated with immune hot. And so this was then also further validated and characterized in a cohort of patients treated with immunotherapy from Caris Therapeutics, an amazing company to work with if you're looking for patients' data and easier access than other companies. Caris has been extremely collaborative and helpful. And so what we found was that 9p loss was predictive of poor response after immunotherapy, but not in the context of chemo or radiotherapy. Again, suggesting that there is a specific role of this event in the context of immune checkpoint inhibitors. With other studies, and in the few years later, there were several other studies that came up. This is just a brief overview of them, where that summarized these studies, ultimately coming together with a picture where 9p loss is a predictor of immune cold microenvironment and also a predictor for immunotherapy response. One important tumor suppressor genes that you might recognize on chromosome 9p is CDKN2A or P6C, which is an important protein that inhibits CDK inhibitors that inhibits the cell cycle, and so it's frequently downregulated or deleted or mutated, mostly deleted in just as a single genes in patients. And so we next ask whether the deletion of simply of CDKN2A or just the region around it was sufficient to determine to predict response. And so in this multivariate analysis, we stratified patients based on whether they contained the arm level loss, so the entire arm, or only the focal 9p21.3 event, or a combination of both. And overall, and this is just one of the several analyses that we did, and overall, what it came out is that the arm level deletion was necessary. So it was really just the arm level deletion that was predictive of this immune cold, at least in oral cancer. And in fact, also looking at patient data sets, what we see is that if we look, instead of stratifying by 9p arm event, we stratify by 9p 21.3 loss, there is no prediction of immunotherapy response. And so right now, part of my lab is really interested in understanding what are the genes on 9p that are critical to promote this phenotype. And it's been a long road. We are still traveling, I would say. But one of the things that we have been doing is that we have been taking a combined computational and experimental approach. So through a computational approach that I will dive into more depth later, we saw that across the 9p region, there are several. It's really not only the 9p21.3 that has this association, but it's really a larger effect, and it definitely spans regions from 9p24 to 9p21 and also to 9p13. And so based on this computational analysis that show the association with the immune score and others, we set out to study these specific regions on 9p, and actually specifically these three, 9p24, 21, and 13. And so what my student Elaine with postdoc Joy also did is that she started from human cell lines and utilized CRISPR approaches to delete large cytoband regions on 9p. These are 10-15 megabases deletions, quite large, and that we managed to ultimately delete. This was the starting cell line that we started from. They're called UMSCC17B. It's an oral cancer cell line, p53 mutant, aneuploid, but as I will show you, no, maybe actually I delete that slide, but stable over time. So despite being p53 mutant, passage in the lines does not change the karyotype. suggesting that either they are not under selective pressure or they are not chromosomally unstable. And so we utilize this approach, which is quite simple, that we call cytoband delete, but it's really simple in the sense that it's just consists of cuts that have to be very efficient on either side, both sides of the cytoband, followed simply by NHJ. And ultimately single cell, these are the vectors that are utilized for the expression of the Cas9, and ultimately followed by FISH and other genome sequencing methods to characterize the lines. And so, so these are the first cell lines that Elaine was able to obtain. And so as you can see, so these are two probes, one the red on 9q and the green on 9p. And as you can see here, one of the 9p probes is missing, while the previous parental lines, as I was showing before, it's present. And again, sequencing of the lines identify the deletion of 9p21. Similarly, we also derive clones that are deleted for the 9p13 regions. In this case, it's about 15 megabase portion of the genome. Again, similar methods of CRISPR-mediated cut followed by single-cell cloning and characterization, leading to the successful deletion of 9p13, and similarly for 9p24. I'm summarizing several years of Elaine's work in a few slides. If you're interested in learning more, we are using the cell selector instrument that has been life-changing for us, in the sense that it allows cells to grow as single clones happier than other methods. And so I'll just, as I said, we're still not at the end of the road, but I'm showing some preliminary data. So we started to characterize these cell lines. And so one of the first things that we did was simply a proliferation assay to see whether the presence of the deletion was conferring any selective advantage or disadvantage. And here I would like to point out that the way that we call these lines is that so we have the deletion clones and the wild type clones. So for each of the deletion clones, in parallel, we derive clones that do not contain the deletions, but were treated in the same way. And what we found through this proliferation assay was that there was no difference in the cell growth of these cells. We then turn to an immunological assay in vitro. These are human cells. We cannot meaningfully transplant them in mice. And so we did this in collaboration with Mark Markovich at NYU, who's an expert in CAR T cell technologies and biology. And he developed this very cool technique that is called peptide-centric CARs. That basically CAR T cells that instead of recognizing a protein on the cell surface, recognize a peptide that is presented on MHC. And in this case, Mark had the HLA-matched T cells that we could use in our assay. In this case was HLA-24. And so in this assay, we plate the target cells, we pulse the cells with peptides. In this case, the peptides is from the protein FOX2B. It's a neuroblastoma peptide. And then we perform the T cell screen. And so here I'm showing the results from the 9p13 deleted and isogenic wild type clones. And so this is the IICD analysis for this T cell killing assay. As you can see, both from the picture here and the quantifications, these cells that are marked with GFP grow happily throughout the time in culture. In this case, they are treated with non-targeting CAR T cells, so T cells that do not express the TCR that specifically recognize FOX2B that is presented. And so in co-culture with these non-targeting T cells, there was no significant difference in the T cell killing. However, in the context of, in the presence of of CAR T cells that are specific to the peptide FOXO-B presented on HLA, we observed a quite significant difference between the 9p13 deleted clones and the 9p13 wild-type clones. So while the 9p13 wild-type clones were really not proliferating, either almost stays the same or slightly decreasing in cell number, the 9p13 deleted clones were happily or more happily proliferating than the wild-type clones. This was a quite significant difference that we noticed in this for these clones, for this deletion that was not present for the others. And we are now, of course, very interested in understanding what is the molecular mechanism, which at least based on preliminary data seems to be independent from the HLA expression on these cells. One other point, in order to proceed into understanding this mechanism, we perform RNA-seq. And so you can see that, so these are the different clones, deletion and wild type for the different cytoband. And you can see that beautifully the deleted clones have downregulation of the genes in the corresponding deletion, as one would expect. In addition to cis regulatory effect, so to downregulation of the genes that are deleted, that are present on a cytoband and deleted, we also observed a lot of trans effect, so effect on genes that are not present on a cytoband. So again, we delete a 15 megabase region, that are 15 or 20 genes there, and we see a lot of changes in other regions. And specifically for 9p13, we are of course very interested now in these regions because of the effect that we saw. We noticed an upregulation of several genes implicated in immune responses, including CD9, CP47, BTCN1, and others. And so we are now proceeding for the functional validation of the underlying mechanism. At the same time, we're also collaborating with Sylvia Gotland at UCSD, who has a beautiful model of oral cancer in murine mouse models. And so we are utilizing some of his cell lines in order to delete the syntenic cytobands of the mouse regions that correspond to the 9p21, 13, and 20. Okay, so now I will still stay on the same theme, but expand from oral cancer to other tumor types with the same idea of predicting and inferring aneuploidies that are associated with immune cold microenvironment and also with poor response to especially to immune checkpoint inhibitors. So in this study, what we did is that we took several data sets, including the TCGA, the CCLE, and the CPTAC. CPTAC is the clinical proteogenomic data set. And by predicting copy number alterations, as well as tumor immune infiltrate through transcriptome or proteomic metrics, we really try to infer which are the aneuploidies associated with immune cold. In addition, we also then built an algorithm in order to predict the specific genes that might function as immune suppressive genes in tumors. So you have already seen this graph before for the head and neck tumors. So this is in a way a similar type of analysis in this case for skin melanoma. And so in this regression model, we want to, so we start from the immune score, the cytotoxic immune score, that we calculate based on RNA-seq, and we want to understand which copy number alteration across the genome predicts the cytotoxic immune score, taking into account after controlling for the overall aneuploidy score. It has been shown, including by also work from this crowd, that high-SIN tumors can lead to immune evasion pathways that are independent of the specific chromosome that is gained or lost. And so here we take into account, we normalize for this compounder. I forgot to say, if you have any questions, just interrupt me at any time. And so this is the plot. So this is the results for skin melanoma. So again, on the y-axis, there is the association for the immune score. So a negative value indicates immune cold and a positive an immune hot. And so our friend 9P 2113 here still scores in melanoma. In addition, we also see an interesting effect for 1Q. These are other examples for other tumor types. So we perform this analysis across all tumors. And this is for lung cancer, where again, 9P is also associated with immune cold, and 1Q gain has also the same effect. Again, here at the bottom, pancreatic cancer, and I'm highlighting here again these two events that we now see across over and over across tumor types, which are again 9P and 1Q. And then finally, here it's bladder cancer, where 9P and 9Q are also strongly associated with immune cold. And here it's endometrial cancer, specifically from the MSI group, so the microsatellite unstable group. Usually these are patients that would receive immediately after being diagnosed immunotherapy, and about 50% of them respond. 50%, which is a very high fraction, but still not all of them respond, and there is not a good biomarker. They all hold tons of mutations, so they have all high mutation burden. And so as you can see here, it was very exciting because there is a direct clinical need, and so for 1Q gains, strongly associated with immune cold. And so this is a sort of a landscape across all chromosomes and across all tumor types. For ARB level SCNAs associated with immune cold microenvironment, and focal level. So here we use smaller events, any type of events, usually at the level of the average is a cytoband level of event that an association with immune cold. And I'm highlighting here again 1Q gain and 9P loss. And here I'm showing in these plots below a similar sort of heat map, similar to the one above. Sorry, where in red are gains and in blue are losses. And in this plot here on the bottom, we highlight, for example, 4 chromosome arms that have a strong signal across tumor types. So 9P loss is here. I also showed you before. 1Q gain is here. And we also have 1P loss and 6P gain that score quite frequently, recurrently across patients. Interestingly here, so here are these are the association with immune cold across different tumor types listed here, and you can see even visually here. So renal clear cell carcinoma is always different than the others. So it's blue when the others are red, or it's red when the others are blue. And it is something that people have observed and speaks to some specificity of this tumor microenvironment. Next, we also looked at specific immune cell types, and so we did that both using the convolution methods on bulk RNA-seq, using various methods, as well as single cell data, mostly as a validation. And so here I'm summarizing the main findings that we see, especially regarding 9P and 1Q. So what we overall saw was that 9P loss was associated with low CD8 and also low B cells. So decreased CD8 and increased B cells were the main associated parameters, and also, but less anti-tumor macrophages. While for the 1Q gain shown here, several tumor types were associated with low level of anti-tumor macrophages, and the signal was stronger than for CD8 and B cells, perhaps suggesting that 9P might act more toward a T cell angle that would fit our experiments that show the difference on the T cell level, and perhaps the 1Q might work on a more on a myeloid type of angle. But these are, of course, just speculation, except for the T cell experiment that's unexpected. So we also validated these findings in a single cell dataset. This is just one example. These are non-small cell lung cancer single cell datasets that were acquired from the literature and harmonized together in one big dataset. It's about 70 patients. And then you see here in blue and red the inference of the copy number alterations through infer CMV, through an optimized method of infer CMV, but fundamentally the method is the same. And so what we can observe here is that there's definitely trends or significant association between the arm level events, the arm level aneuploidies that I just described to you, and immune effects. So for example, CD8, so for example, 9p loss and lower CD8, which is frequent across tumors and validated in single cell lung, but also skin tumors, as well as also trends for 1q gain and anti-tumor phages. Okay, so in addition to doing all these sort of pan-cancer, pan-chromosome regression models, we also wanted to dig deeper into the specific genes. And so in this case, we built a machine learning model based on predictive model based on so this model is based on several parameters. Each gene is assigned several parameters, including frequency of gains and loss and deletion, but also gains, correlation between DNA and RNA level, correlation between RNA and protein level, as well as the association between the RNA expression level and the T cell infiltrate, so the immune score. And as a positive control, so here there's not so much known about the immune regulator. There's a lot known, but in reality, if you want a training set, it's still limited. But we use the CRISPR-based screen, where people have performed CRISPR screen, either CRISPR-I or CRISPR-A, mostly CRISPR-I or CRISPR-cut screens in cells, and I've injected the population immune competent, immune compromised mice, and I've seen which genes confer resistance or growth advantage only in the presence of the immune system, or only in the presence of CD8, if it was an in vitro assay. So I won't go through all the different models because of time, although I'm happy to tell you more, and this was part of the few slides that I added then. But in the end, we chose this random forest model. It's behaved slightly better than others. And overall, these are some of the predictions of the identity, so the location of our predicted genes on the genome. This was done in this and plotting the sort of pan-cancer analysis. As you can imagine, the positive controls are not available for every tumor types. These screens were done in available cancer cell lines. So, but anyway, so these are the predicted immune suppressor genes and immune oncogenes, as I call them. And so these are the location in the genome. And so you can see that there is a good numbers of them of the immune oncogenes on 1q, consistent with the fact that 1q gain is associated with the immune poor in our data, in our analysis, on 9p as a loss, and on 20q as well, and 18q. And finally, this is a gene set enrichment analysis that we did on the predicted genes. And interestingly, we find that, perhaps not surprisingly, several pathways that are known to be whose deletion is known to promote immune resistance. For example, genes implicated in antigen presentation, in attraction activation of T cells and of macrophages as well, and apoptosis itself. So whose deletion really promotes resistance of different, probably at different stages of immune recognition and killing. And then on the other hand, for the immune oncogenes, so for the ones that are overexpressed or, grained, the pathways that came up were cell cycle, RTK signaling, mRNA translation, and immune suppressive cytokine. Some of them are quite interesting and why mRNA translation would be there. I don't know, but I think it's an interesting hypothesis that one could potentially test. But we have not done that. Okay, and so one of the final steps of this effort to identify the genome-wide and tumor type-wide genes that whose deletion or gain promotes immune evasion was to cross this with a data set, MSK impact data set from MSK, where patients were sequenced based on their impact panel, and that were also followed after immune checkpoint inhibitors. And so this is just one example where here I'm plotting on the y-axis, not the association immune score as it was before, but the survival of the patients based on whether they contain gain or loss. And so this is a high value would mean worse survival, and a negative value, so I see it as a HR lower than 1, better survival. And so first of all, you notice that all the curves are above 0, so they tend to predict worse survival. That's not surprisingly. And there's definitely a big peak here at the level of our favorite 1Q. And so we are very interested in understanding, of course, what are the mediators of this 1Q. And you can see this here also in this survival plot where patient stratifies based on the presence of 1Q or no 1Q are different in terms of survival after immune checkpoint inhibitors. Okay. So I said that was one last thing, but this is the last. So still in the context of this study, we decided to collaborate with Ben Stenger at UPenn, who has a little bit of a mouse TCGA that we could put our hands on. So he basically started from a KPC pancreatic mouse model, where you start from one cell that gets a KRAS and a p53 activation. And then he derived several single cell-derived clones. Each of them are different, and then implanted each individual cell back in a tumor. So we got these cells, and in collaboration also with INI, we reimplanted them and measured the tumor microenvironment as well as their copy number alteration. And so I'm calling it the small mouse TCGA because then we basically did the same thing that we did for human on this small 10 cases data set. And so here is the analysis. Sorry, I realize it's a little complicated, but basically the first plot is just the association between the copy number alteration. Here we are not splitting gains and losses because with 10 samples, we just do correlation, Pearson correlation, Sperman correlation, in the sense that we would lose a lot of the values. Plus, these are pure cell lines, so we can really precisely calculate. There's no problem of purity, which instead, of course, happens in human tumors. So this is the association between the copy number and the CD8 T cell, and so in the mouse genome. And so in the bottom 3 plots, now everything is plotted on the human genome. And so here, basically, what I wanted to point out, so this is the mouse genome plotted on the human. This is the human data that I showed you in the previous slide. And now this is the mouse plus human, mouse per human. It's the product of their values. And so positive values would mean the same direction for both mouse and human. And as you can see here, this was for the association to T cell. As you can see here, our chromosome 9p scores quite nicely in this mouse data set. And here, sorry, here I'm realizing that I have not extended far enough this plot, but here you can see that this is the same analysis for macrophages. And what you can see in the bottom plot that really shows the basically the associations between the mouse and the human, because it's the product of the R values on the mouse and the human, so it's positive when they both go in the same direction. and there is a big peak on the chromosome 1q of the human, suggesting that those regions are the same. And of course, one can do much more, and we are diving deeper into it in the sense that thanks to the lack of complete synchrony between the mouse and the human genome, one can also try to assess what are passenger and what are drivers. Okay, this was for the first part, but for the second, I think I will try to squeeze it because it's getting late. But do you have any questions on this part? Yeah.
I noticed that your 5q deletion is being highly associated with in the small.
That's true. 1q, yeah.
5q.
5q, sorry, yeah.
5q deletion. Specific pathways that was associated with other groups.
No, but it's definitely something. It's definitely one that we have seen over and over. So we are quite confident about, almost similar to the 9p.
So one of the most common alterations, 53 mutated complex karyotype leukemias. So
yes,
we're interested to understand what genes on the interval are important for that.
Yes, absolutely. Yeah, no, yeah, it's definitely one that is not just one case, but it's something that we have seen over and over. Great. Yeah.
For your immune scoring, you were talking about looking at the infiltration, but can you also look at the activity of the cells?
Yes, I just showed one part of the graph. Yes, and in fact, there are some aneuploidy that are associated with increased. Sometimes we see the same event. For example, for an NP, that is loss is poor and gain is hot. Not always, because sometimes one event simply does not happen, maybe because it's strongly selected against. But yes, we haven't shown it, but yeah. Yeah.
This might be, sorry if you went over this, but you know, when you're talking about your immune score and you essentially take into account their chromosomal stability across all.
Across all chromosomes, yeah.
Do you ever explore interactive facts which you may, that might be present as well between just overload and specific?
Got it. Yes. We have started to do that with a mutual information type of approach, actually through somebody who was actually a master student here and now is in Germany, trained here in statistics. So we're trying to use this mutual information, which is supposed to be more potent than simple correlation, to try to address exactly that. Yeah.
Have you ever looked for the information? Have you ever looked in the other direction? Like things that have more immune infiltrate.
Yeah.
Sorry, did you just say that?
Yeah. Yes. I mean, for kidney, as you also know, for kidney, often where you have immune evasion for some tumor, you have immune pot for the others. That we still cannot explain why that might be, but is there. Yeah. Okay. Great. So I just don't have a lot of time, so I will just go through it relatively fast. So we recently developed a method to induce chromosome-specific gains and losses in human cells. This method can be used to generate clones with chromosome-specific aneuploidies. We now actually have a CaryoCreate 2.0 version. So if you're interested in it, you can just talk to me or write to me. I'm happy to share this method or the advanced version. And so for this part of, for this project, my student Alia was interested in undoing the aneuploidy that is present in tumor cell, as also others have done in the field, including Jason Schelzer and others. And so we basically started from for chromosome 7K, which is the most frequent event across all solid tumors, including brain tumors, where it's extremely frequent, but also colorectal cancer. So we started from a cell that has chromosome 7K, and then we use CaryoCreate to get rid of the chromosome 7. In this case, in addition to chromosome 7, we also got rid of a chromosome 5 trisomy. For a chromosome 5 trisomy, we didn't really see much effect, both in terms of growth in vitro and in vivo. However, for chromosome 7 gain, we saw a very dramatic effect in the growth of the tumors, especially after implanted in vivo, in mice, in this case, is subcutaneously. And so one of the things that we asked was whether EGFR is a chromosome. So there is no HEC DNA here, by the way. no IICD, no complication. So that EGFR is on chromosome 7. So one obvious question is EGFR the driver, the only driver, and are these trisomy 7 cells more sensitive to EGFR inhibitors? So I'll answer that specific question in a second, but in order to characterize the drivers on chromosome 7, we integrated several data sets, including data from cancer cell lines and patients, and our own CRISPR-I screen. And so what we identified was about 10 oncogenes on chromosome 7. So chromosome 7 is really a chromosome that is full of oncogenes, as it was alluded before. But the question is, can we really understand what they are doing cooperatively? And so 3 of the oncogenes that don't need an introduction are BRAF, EGFR, and RAC1. So as I said, there are many more, but for the purpose of this project, we focus on these. And so what we saw was that, as expected, in the chromosomal loss, in the trisomy loss. So when I say chromosome 7 loss, is trisomy loss. In the trisomy loss cells, there was an expected decrease of the RAC1, EGFR, and BRAF here. And this is also correlated with the type of loss. So here, one thing I didn't say is that we also have 7p and 7q cell lines. But basically what we were able to observe is that the decrease of about 30–40% of dosage was observed in these isogenic lines, as expected. And so when we then looked at the dependency map for chemicals, for drugs that may associate in their dependency score with a chromosome 7 gain, specifically in colorectal cancer, what we saw was that EGFR inhibitors, for the most part, were not really scoring in this differential sensitivity analysis, while the MEK inhibitors were clearly at the top. So perhaps not surprisingly, in the context of an increased dosage of several genes that participate in this RTK MAP kinase pathways, and also RAC1 that has also additional function, EGFR in itself is not does not lead to a specific sensitivity to the cell. However, MEK inhibitors do. So trisomy 7 cells are more, they die more, are more sensitive to MEK inhibitors as compared to colorectal cancer cell lines that do not contain the trisomy 7. And so our hypothesis was that perhaps the synergistic increased expression of several genes on the pathways, as I just said, was leading to this effect. And so we are still in the process of fully finalizing the mechanism, but in part we did that. So what we did was that we knocked down with CRISPR-I, and CRISPR-I in this case, just mediated by DCAS9, allows for a mild decrease in dosage. And so we took the trisomy cells, we knocked down individually or cumulatively or together, BRAF, EGFR, and RAC1. And as you can see, the decrease, it's really on the order of 20 to 40%. And in this case, we measured the doubling time of the cells as a measure of fitness of the cells. And what we saw was that while the individual genes didn't really do too much, they did some, but not too much, the doubling time of the cells, it's really the downregulation of the 3 of them together that had a synergistic effect on the doubling time. Again, suggesting that a small decreased dosage on 3 genes has a pretty significant phenotypic effect. And then finally, we also use our cell lines. So control is trisomy and all the different 7q loss and 7p loss to validate some of the inhibitors. And we were able to validate these MEK inhibitors as being more sensitive in the trisomy 7 as compared to the disomy 7 cells. RAC inhibitors as well, but no difference in EGFR. Yes, one last thing that, sorry, I don't have a slide about it, but we are also collaborating with MSK. They have a trial of colorectal cancer patients treated with a combination of these inhibitors, in particular MEK inhibitors with BRAF inhibitors, and chromosome 7 seems to be a biomarker for good response. The numbers are still small, but it's suggested. Okay, and so to conclude also this part, our lab is very interested in developing tools to model aneuploidy, and we have done so in human cells, including in this colorectal cancer model, where we dissected the chromosome 7 role. And we are very interested in doing that in other models, including brain tumors and actually leukemias as well. Great. And with that, I'd like to thank my lab, especially Elaine, who's really the master of the 9P project, as well as a former member of the lab, Alia, and as well as Iman and Manjunata, who are further developing and improving the CaryoCreate method. And thanks all the funding until they're still there, and collaborators, Scott, I mentioned Scott, yes, main collaborator for this topic. Thank you. Yeah.
The 1Q association with maybe macrophage infiltration. CL1 is the anti-apoptotic protein, similar to BCL2 is in the 1Q interval. It's also, I think, very important for phage function, but how that would relate to amplifications on a particular tumor as opposed to a macrophage. I've seen some people speculate about mitochondrial transfer between tumor cells and other tumor microenvironment. So it's possible that mitochondria with high MCL1 are being transferred or traded with phages in the tumor.
Yeah, 1Q is the biggest chromosome of our genome, and our chromosome, and MCL1 is there, and DM4 is there, and so there are several genes that have been already that have been already proposed as being oncogenes. That doesn't mean that they're necessarily working to explain the phenomenon that we see here. So to be honest, I don't know the exact drivers, and I also doubt that. I think we might be missing it. We might be in the end missing it if we are just using computational methods. So I think we ultimately, with a certain range of prediction, even loose prediction, I think ultimately a screen is necessary. Of course, deleting 15 megabases of genome is easier than introducing 15 megabases of genome, because there are also gene clusters that for which you might need to amplify them.
How efficient is, sorry, if I answer the question, how efficient is your 15 megabase deletions that you were mentioning earlier?
So, I mean, I would imagine that it's cell type dependent. In these cells, I think we were quite lucky. It was 9% as a population level measured by FISH. I didn't expect it to be so high. We tried for years to do it through homologous recombination.
That's what I was surprised.
But it did not work.
Okay.
The cells are not embryonic stem cells are not, they don't like to do HR. That's I don't have a better explanation. But we actually did several tests, and with the efficiency, the preferred pathways is 9 to 1. Is that?
I don't know if you can hear people
Yeah.
on the Zoom. Okay.
2 questions. What are your thoughts on the presence of aneuploidy cells in normal tissues? Does the phenotype seen in cancer cells also explain why it's present in normal cells? What is kept in checks? And 2, what are your thoughts on using primary aneuploidy cells for studying phenotype as opposed to engineered cell lines?
Oh, interesting questions. Yeah, I think that the presence of a small portion of aneuploid cells in normal tissues probably carries some of the same phenotypic advantages and disadvantages as well. Meaning it's selected during the tissue and during the tissue homeostasis and somehow retained. And generally is limited to 1 or 2 or a few aneuploidies. So I think what we're missing in the normal tissues, thankfully, are complex karyotypes. Now, how do you go from from a normal aneuploid cells in a normal tissue to complex karyotype, and what ultimately leads to the transformation? I think it's a question. Regarding using normally occurring aneuploid cells, I never thought about that. It's an interesting idea. I mean, in theory, I think that when we have an hypothesis of biological mechanism, we should be able to recapitulate it by engineering the aneuploid. If we cannot do it, it's mostly because of technical limitations so far, but we might be able in the future.